we are very happy to have Fritsche here. She is a professor at the, I just had it, Leibniz Research Institute for Developmental Medicine in Düsseldorf. It's a joint appointment with the RWTH in Aachen, I think. RWTH? Yeah, but anyway. So we just stay with Düsseldorf and the Leibniz Institute for Environmental Medicine. So she's very well known for her work she's been done or had been done on skin and brain development or metabolism, and especially with brain we had also a collaboration in the past, a couple of years ago, with neurosphere development. And we already heard in our previous talks today about the in vitro battery on developmental neurotox, which was also an EFSA project. And today she will tell us more about an in vitro battery addressing developmental neurotoxicity. So thank you very much for coming, and we are looking forward to your presentation.
Thank you very much for your kind introduction. So I used to have an appointment with the RWTH in Aachen, but for many years now I'm with the University in Düsseldorf in a joint appointment with the IUF. But first of all, I would like to say I'm really happy to be here today, and especially for this birthday of the BFR, because when I returned from the United States from my postdoc there, the very first money that I received for the model that I will also talk a little bit about today was a grant from CEBET, from the BFR, so it was Horst Spielmann at that point in time. And after that, the model got also promoted by an initiative from the BFR by Andrea Seiler, and we had a BMBF project together. So I have a very, long-standing good relationship with the BFR. So, but first of all, I think I have to talk about my conflict of interest, because since March, I'm actually the founder and co-stakeholder of this company, the Entox. So we are making in vitro methods available to the public, but I think I have to indicate this now. Okay, but now let's talk about DNT. So how did the whole DNT work start? So it was really Alan Goldberg from CAT, so it was the one for Thomas leading CAT, who had the idea to bring DNT onto the table, and there were great workshops there that kind of laid the ground to the first international stakeholder meeting, ISNET meeting on DNT, where a concrete roadmap was created in Europe. At the same time, also the EPA, the Strategic Research Action Plan, DNT was actually a part of. And I think the real work that I'll represent started then at the 2016 meeting in Brussels, that where DNT's attention got to the OECD, and at the joint OECD-EFSA workshop, where it was decided that there is data requirement for DNT, the in vivo is not able to cope with this data needs, and that a battery should be assembled and methods are ready to do so. And this is when it really all started off. And since then, mainly EFSA here, the EPA in the US, and also the Danish EPA have been sponsoring the setup of this battery. So how does the battery work? So it's not a one method replaces one animal experiment. We all know that by now. So it's really coming from basic biology of brain development. So for the brain to develop, it's fairly complex. We need a lot of different processes that occur in a also time-orchestrated manner. And all these different processes now have individual in vitro methods set up that all together build the battery. And I just want to get to you an overview, so don't read at every single point individually, but these are the assays of the battery that are in the guidance document that was mentioned earlier, and I will also come back to it later. So basically you see here, is this ready for pointing? Yeah. What you see, these are the different endpoints that I showed you on the slide before. Then you have the institutions involved in this work. So you have the sign from the EPA, then it's Marcel Leist from the University of Constance, so CAT Europe, and in green the IUF. So these are our assays. And basically it's pretty obvious. So for one thing, they are mainly human, but not all human assays. Then for some endpoints, we only have one assay. And then there are endpoints like neurite outgrowth, where there are really multiple assays available. So this is the current state of the art, because a common set of chemicals were tested across these assays, which had a certain readiness at the point in time when we started. But it's an open game, so it's not set in stone, because as science moves on, also the battery will move on. And we have seen this actually 2 times before from Maurice, and also in a modified form from Nicole. This is a great paper coming out about the scientific confidence in NAMs. And looking back now into what we've done with the in vitro battery, we've actually been addressing a lot of these points. And I will especially go for the human relevance now in the following slides. Fit for purpose will be addressed by some case studies. Technical characterization we actually do have in tox terms, and also data transparency. So all the first data from our first testing that was sponsored by EFSA and the US EPA is now can be found in the ToxCast database. So it's open access. Now, I'll start with one example, so with our assay of the battery, just to give you an insight, so what are we really doing there, kind of to fill the first layer with a little bit of meat on the bone. So the assays that you saw on the slide before, which are ours, are all based on these human neurospheres. And these neurospheres, when you plate them just upon their free will, they start migrating, and there's a lot of cell-cell contact and communication going on. And when you stain these cells after 5 days, so it's pretty quick for neurodevelopment, you see that there is a mixed culture of neurons, astrocytes, radial glia, and oligodendrocytes, which can be quantified. So it's important for these DNT battery assays that they really develop in vitro, and we can show that the cells keep on migrating over time. And also you see these are the first radial glia cells, and they continue to differentiate. So these are the 5 days that I've been talking about. And so this is just an example of so the very basics of the assay. So all the assays of the battery have done their homework on this part. And how do we identify cells? So this is a bigger image of the whole neurosphere. So if you imagine, so we do screening. So each well of a 96 well plate contains such a beautiful neurosphere, which I personally is always fun looking at. And you can also imagine that not the students can count the cells anymore and measure migration. So this is impossible. So we decided to go for an artificial intelligence approach. And what we actually do when you zoom in, our high content imager finds the nuclei, so that's easy, but how can I teach a program to identify a neuron or an oligodendrocyte? This is much, much harder, because the cells in such a primary system, they do not all look the same. So you really have to train them. So in what we did many, many experiments, did human hand annotation, and in this example, it's the oligodendrocytes. So the lab was really happy when this task was over, I have to say. And then the AI was trained, and we looked for the true and false positives and negatives, and then compare the results, run with new parameters, so that in the end, we have now an AI that analyzes all the plates that we're running. Moreover, we're really interested in scientific validation, so to make sure the morphology of the cells is really like a brain cell. I mean, a neuron should look like a neuron, right, and should express the right markers. It should develop over time. Like this is similar to the migration, the percentage of neurons in the culture goes up. Then the responses to physiological stimuli is really important. I mean, if you claim you have a neurodevelopmental assay, the cells should better respond, as kind of like in vivo on the physiological side. And here we treated the cells with DAPT, which is a notch inhibitor, and notch is a very strong pro-neural stimulus in vivo, and you can see that also the number of neurons in our cultures really go up. And when you activate the role pathway, like here with narciclazine, and this is the quantification, then the neuronal number goes down. This is just an example, and more or less all the assays of the battery have done this, and you can find all this information in actually the tox temps. I should be on the next slide, yeah. So these are templates developed from the OECD guidance document 211, specifically guided to such in vitro systems, and all the assays in the battery have filled out these tox temps. It's quite some work, so there's a lot of information in there, and they are freely accessible with the guidance document. And I think this is really important also for data transparency and also for reducing uncertainty, that you really understand your test system well. Now, next thing is, when we do all this testing in AI, everything is fine, but what comes next is data evaluation and, in the end, classification. And so we really spent some time on setup of a biostatistical pathway that is pretty much fit for purpose for such a, I would say, more variable 3D methods. And actually we did compare different statistical approaches to this, and submitted the work, but at the, now I went forward. But here a preprint can be found at the bioarchives, because I think it's really something that needs to be looked at when looking at NUMs and classifying results from NUMs, that you use the right statistics, because statistics can really change the outcome of the methods. So in the end, our classification model understands if there's no hit, that's pretty easy. But what is a specific and a non-specific DNT hit? So the specific hit is when the cytotoxicity clearly distinguishes from the specific endpoint, like migration, differentiation, and so on. The unspecific hit is that the confidence intervals of the benchmark concentrations overlap, and then there are borderline cases. So and this classification has been used then also for determining the predictive capacity of the battery, and I have to say this is only the EU part of the battery, so it's only from Marcel Leist's lab and my lab the data. It's lacking the EPA data, because at that point in time the data was not ready to be evaluated as a whole, but we are working on that at the moment. And what was very critical and is still critical is the debate, what is a DNT positive? So that's very, very much different from the skin sensitization, where the database is much more clear also for humans. We don't have this luxury in DNT, but there are several manuscripts that have dived deeply into this topic, and we came up with 46 chemicals. We said we can be pretty sure that these are really the right positives and negatives. And so our sensitivity and specificity is actually quite good, I think. You might wonder about the sensitivity, but this was not a surprise for us, because when you look at the endpoints of the battery, so the ones of you who are familiar with neurodevelopment have recognized that, for example, myelination is not in there at the moment. We don't have astrocytes endpoint in there, and also the microglia are missing. So, and plus, this is just the EU part of the in vitro battery, so the neuronal network formation is missing. So, and when we had a smaller data set, including the network formation, then the sensitivity immediately goes up a lot. So I think it's the right way to go. We are aware of the gaps. They are also in this guidance document, and this is the provisional guidance, so it's actually now pretty much done with the second phase of commenting at the OECD level, which was mentioned earlier, and I hope it will be then published at some point next year. So the basis of this guidance document is the data that I showed you, what the EPA and us in Europe produced with the battery, but also there's a little bit more to it, and I would like to go into the case studies briefly with you. So the first case study was guided by EFSA, and actually EFSA asked the question on hazard assessment of two pesticides, deltamethrin and flufenicet. And well, we had the data from the battery, and you see the outcome here. You see the little US, that means it's an unspecific hit, so it's not really the highest concern was at that point in time. Then the little s tells you it's a specific hit. And then we have in red the 3 specific hits for delta methrin, where 2 of the endpoints responded with at fairly low concentrations, and all the other assays of the battery were actually negative, which is not surprising because we don't want every assay to be positive with every chemical. In contrast, the flufenostat didn't give any alert, so that was kind of the negative part of it. And based on the data of the battery, plus actually a systematic literature review of all in vivo data and other in vitro data, and also a guideline study that existed, the EFSA working group set up this AOP network on the key mechanism of delta methrin pyrethroid, so it acts on voltage-gated sodium channels, including here the altered neural network function and the oligodendrocytes endpoint into this AOP network. And so this was part of the AOP-informed IATA. So the second case study, this is a different one, and this is coordinated by the NTP, and Nicole in the discussion touched briefly on the flame retardant, so this is actually the case study that she was talking about, and here circled are the new better replacements for the bad flame retardants up here. There are 2 examples, BD47 and TBBPA, which are already phased out, and when you look at the pattern, so these are all the different assays of the battery, plus the zebrafish is also in here, of the EPA, the IOF, and Constance. You see a very similar pattern of concern compared to the phased out ones. So I'm not really sure if it's a really better replacement. So, but this is a case study how you can work on compound classes. Okay, and then as the last part, I would like to show you another way how to increase confidence and probably reduce uncertainty with the DNT nums. So we've been working on combining transcriptomics and what we call phenomics. So phenomics is really the high content imaging because we're looking at phenotypes and build up AOPs from there. And I brought you 2 examples. So in the middle, you have the neurosphere assay, where the beauty is that you have multiple endpoint readouts at the same time. So you run one experiment and you get lots of different endpoints with the AI. And so there's one example. The green guys, again, are the oligodendrocytes compared with RNA-seq data. Made us build up an AOP network around oligodendrocyte differentiation and maturation, in the end acting on myelin. And the second case study is a different one. Oops, there it is. It's actually based on a very strange migration phenotype that we observed. And we could build an AOP that's concerned with cell adhesion and migration. And we actually, when we built this AOP, we actually worked with one compound, and I kind of thought, well, I'm probably never going to see this phenotype again, right? Because it was a natural compound, not really a chemical. Well, and it happens that, in our screens, all of a sudden, we had exactly the same phenotype again. So even these rare phenotypes might show up again, so it was a nice confirmation of the AOP. Okay, so now where's the battery standing right now? We've heard a little bit about ASPIS from multiple speakers today, although, Bob, you didn't really talk about ASPIS today, at least, there it is. So, so the DNT assays from my lab are in the ONTOX project. It's a completely different approach with starting from human ontologies and, kind of leaving the rat on the side. So really a different approach to risk assessment. And Marcel's assays are in the Risk Hunter project that Bob coordinates. Then all the assays are challenged with compounds from the US NTP, so they're really putting a lot of resources in this right now, which is great. Then the assays are also part of the PARC project for actually investigating compound classes and taking the assays further, complementing the battery, closing the gaps. And also the assays are used in the endpoints project, so that's part of the urine cluster, for bringing the endocrine aspect into the DNT. And we found roughly 10 nuclear receptors responsible for guiding different parts of migration, different parts of neurodevelopment, and so taking this also to a different level. And with this, I would especially like to thank Andrea Teron, because he has done an outstanding job of supporting the whole battery, and of course Tim and Marcel, our testing partners, and also the JRC, the Danish EPA, and the OECD. I mean, they have been really in total support. And Kevin and Bill retired from the EPA, but although they are retired, they are doing a really great job of writing the guidance document and dealing with all these comments in the reporting. And last but not least, my lab and the multiple sponsors. And thank you very much.